Welcome to Datta Biology class. Let us start a conversation on DNA replication. This is the concluding part. Till now we have concluded this one, that is conversion of triose phosphate to monophosphate. How? Some adenine, sugar, phosphate. This comes to the stand and they releases these two pyrophosphate. These two means pyrophosphate, and pyrophosphate on hydrolysis produces inorganic phosphate and energy. That energy is required for replication. Now this is a replication bubble, this is a bubble, this is a bubble. So bubble, if it starts from here, so there will be the end of this, and it bubbles from here. It is bidirectional. Here also it can start by. And bacteria and others, they are unidirectional. Now, if this bubble forms a fork. In case of elongation, I am telling. This forms a replication fork. Replication fork is a fork shaped. So suppose this is 5 prime, this is 3 prime, and this is 3 prime and 5 prime. Now, conversion of this has converted, these are coming and they will join. That is, for primer, which some primer will come here, some primer will come here first, and thereafter the strand will go. That is elongation of new strand. Who elongates this? The elongation is done by DNA polymerase. This elongation is done by DNA polymerase. There are 5 types of DNA polymerase. Polymerase 1, 2, 3, 4, 5. DNA polymerase 1, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, There are 5 types of DNA polymerase in bacteria. Let me say that polymerase, there are 5 types of polymerase. In bacteria and E. coli, this polymerase 3 is the main elongating enzyme. Here, this is known as replication fork, and from here in bacteria and others, polymerase 3 in human cells also. Polymerase 3 is the main enzyme. So here the primer first attaches itself, which I have told you earlier. This polymerase attaches here, and DNA polymerase, until and unless primer attaches here, DNA polymerase cannot act. So DNA polymerase 3 start coming here, taking the triphosphate. So taking the triphosphate, suppose it is A, triphosphate will come at T, the T triphosphate will come here, along with sugar and phosphate bond, it will start from here itself. So there is a single strand it is coming. From 5, it start DNA, it can act from 5 prime to 3 prime. That we know earlier also, even then today. So it starts from 5 prime and it one primer is enough to continue this. So it continues. This is known as leading strand. This is known as Leading, leading strand. Clear? So leading strand will follow, elongate. This will elongate. The new strand will elongate from 5N to 3N in this way. And it requires a single primer, and the strand is continuous. Number one. DNA 3 does it. And what are the functions of this? DNA polymerase 1, 2, 4 and 5. They are meant for repair and error checking. The other DNA polymerase 1, 2, 4, 5 mean for repair and error checking. If there is any error, it checks and it repairs. This is the function of the other polymerase. In E. coli, DNA polymerase DNA polymerase 3 is the main enzyme. In eukaryotic cells, there are other enzymes. 1, 2, 3, 4, 5. There are polymerase DNA, polymerase alpha. In eukaryotic cells, DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon, EPSILON. These are the three primary polymerases which are responsible for this elongation of the strand or forming the new strand. So, leading strand proceeds from 5 to 3 here. In E. coli and others, in humans also it is written in the book that DNA polymerase 3 is the main enzyme, while the DNA is, you should write in that way that DNA polymerase 1, 2, 3, 4 and 5 are meant for repair and error checking. And in case of eukaryotic cell, DNA polymerase alpha has been identified, DNA polymerase delta and DNA polymerase epsilon. These three are the elongating enzymes. Of course, after primer comes here, RNA primer sits here, they can work. Okay, this form the leading strand. Now, lagging strand. Lagging strand we know that is, this is 3 prime, this will be changed a little bit. 3 prime to 5 prime, okay. This is 5 prime and 3 prime, it will not start from here because DNA polymerase, what may it be DNA polymerase alpha, DNA polymerase delta or DNA polymerase epsilon, may it be anybody, it cannot start from 5N. So, here it comes and it starts from RNA primer 6 here. Again, one RNA primer indicates that you should start from here itself. So RNA primer sits here, RNA primer sits here, RNA primer sits here. So this is, now it become, here 5 prime, here is 3 prime. It is 3 prime, 3 prime, 3 prime to 5 prime. RNA polymerase will say that 3 prime. So it starts from, this is 3 prime, it's 5 prime to DNA starts from here. So here is single strand. Again, DNA polymerase will come and sits here from 3 prime to 5 prime. It will go in this way. Again, it will go in this way, forming a number of strands having gap inside. Why this gap? Because this RNA prime, this DNA polymerase cannot join this. These are actually called nicks. These are actually called nicks. Nicks. These are called nicks. Okay. These will be done by another enzyme known as ligase. Ligase will, it acts like a glue, or it joins the sugar phosphate bond. This is Nix is formed by ligase. It acts like glue, it joins it, it makes the sugar phosphate bond. It joins here. It again joins here. It again joins here itself. So in this way, these nicks are filled up by ligase, L-I-G-A-S-E, ligase enzyme. It is a replication machinery enzyme. This ligase enzyme fills here and it forms the sugar phosphate backbone of the lagging strand. This is leading strand, this one is known as lagging strand. This one is known as lagging strand. Lagging strand from 3 prime to 5 prime it goes, but this lagging strand starts its replication from 5 prime to 3 prime, from 5 prime to 3 prime, from 5 prime to 3 prime, from 5 prime to 3 prime. In this way it goes, because here it should have 5 opposite 3, here 3 opposite 5. So although the lagging strand cannot start from 5 prime, but the lagging strand actually RNA primer sits in such a way that it starts from 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime, and again here 5 prime to 3 prime. So it will be adjusted. Clear? So in this way, the leading strand is continuous and lagging strand is not continuous, but there it forms a mix in between the several fragments called the Okazaki fragments named by after its discoverer, and these are called nicks. The nicks are filled up by a glue-like enzyme known as ligase, and it forms the sugar phosphate backbone of the lagging strand, and ultimately the lagging strand becomes continuous. So it forms both sides. So we have seen that this 2 has been formed and again this 2 has been formed. So it replicates. Replication, when it is over, what will happen? Still, we have not completed the replication. What happened? Let me say, after elongation, I am erasing it one. Leading strand and lagging strand has been formed, and those have been joined by ligase. So ligase is another enzyme. So here elongation will come DNA polymerase and ligase. These are the 2 enzymes required, and DNA polymerase has been 1, 2, 3, 4 that you will write. And in case of eukaryotic cells, DNA alpha, DNA delta, and DNA epsilon. These are the primary polymerase enzyme for elongation of the chain or elongation of the new strand. Now, termination. Now, once it is done, then it is termination. Termination phase is over. So it is termination. You write here And coming, 6 you write, number 6 point is termination. Somehow it has to terminate. So termination is done throughout the entire length of the chromosome. Throughout the entire length of the chromosome, the new strand has been formed and it terminates the replication. Even if it terminates the replication part is not over. Replication part is not over. How? Because the links are joined by ligases and other. Even it is not over because in the cell, when it is going on in the cell, in the nucleus, when these are duplicated, there remains so many RNA primer. So the leftover RNA primer should be removed, as in case of any machinery work and in other works we have seen that the debris or roughage, the debris has to be removed. So whatever RNA primers are there, that has to be removed. That removal is done by, you write it in termination, or you can give subheading, removal of, right, removal of RNA primers. Removal of RNA primers. This has to be done because once you have taken the charge to change the computer to identical computers in a office, so you will have so many roughage or debris that has to be removed. Who will remove? So there will be another 2 enzyme that you write, another 2 enzyme. If EP or something, a minute, FEN1, FEN1, number 1, number 2, RNAH, RNAs H. These 2 enzymes you should indicate. This is known as flat endonuclease. FEN, write it, flat endonuclease. Flat N-U-C-L-E-A-S-E. Flat endonuclease and RNase H. These are the 2 enzymes of the replication machinery who removes the RNA primers used for elongation. Still it is not over. Still the replication is not over. How? Still the replication is not over. How? In chromosome, you have come across in your smaller classes that in chromosome, 2 chromatids are there and 1 membrane is there, and the end is somewhat full of DNA. Why it is made? Because in crossing over, and chiasma formation in crossing over. So they exchange their parts. Say from these parts they exchange, these parts they exchange. So the end of the chromosome has a repetitive DNA sequences. How? I will tell why that, because it protects the chromosome, another chromosome fusing with this. In chiasma crossing over, sometimes it may so happen that some other chromosome parts may join. So they make some repetitive DNA sequence. What is repetitive DNA sequence? Like this. Adenine, guanine, thymine, cytosine, adenine, guanine, thymine, cytosine, like this. So these are repetitive in nature here. So it becomes, this is known as telomere. Telomere. In the chromosome we find at the end a portion which is more concentrated or condensed to form a protective cap-like appearance, which is meant to protect this chromosome from joining with other chromosome parts during replication or during cell division, crossing over and chiasma formation. They protect this, and here they make so many repetitive AGTC, 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 and they repeat in such a way that it becomes very super quiet. So DNA ligase, DNA polymerase cannot find this. Then what will be the exact nature of this repetitive sequence? That is done by one enzyme known as telomerase. Telomerase, which forms the repetitive sequences of the nucleotides. So the repetitive nature of sequences of the nucleotides are exactly same position has to be done because it is a case of replication. So DNA replicates, the DNA here, it replicates because it is easy. AGTC, this is easy. And if it contains a repetitive DNA sequences, here nucleotide sequences repetitive, that cannot be done by DNA polymerase alone, because DNA polymerase is a simple machine. That machine cannot do such a fine work. So fine, superfine machine is required. That superfine enzyme is known as telomerase. So we have got another here, that telomerase has come to make the repetitive DNA sequence of the telomere of the chromosome. So it is now, still it is not over. Still it is not over. How? Because once the chromosome replicates, the last part I am coming, once the chromosome replicates, suppose this has been formed. Here AGTC, AGTC. These are all nucleotides with sugar and phosphate backbone, original. And the next one has been formed. I am writing it in different color. TC, AGTC, AGT, IGT, C, A, G. So this is formed by other sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. Okay. So another has been formed. Here also, in this case also, another has to be formed. So another formed here. These also formed here. So new strand, new DNA replicated form is there. This is a new replicated form is there. But it is not over. How? Because it has to be in the original position. Original position, it was coiled. It was coiled like this. Is it coiled? No. So this has to be recoiled because once you have taken the order or you have got the tender of doing this replication, you have to give this in the original form. So that has to be recoiled again. So this finally you should write there once it is done, it is not over, it is recoiled. Coiling was there, in this way coiling was there, coiling was there, so it is recoils, recoiling, C-O-I-L-I-N-G. Recoiling takes place or helix. You know, the new strand coil as the new strand coil in the form of a double helix. We write that new strand forms, I am writing here, new strand coils, new strand then coils with the old strands and form helix. This is the last point you should write, that once it is original position helix, so it is like a double helix, new DNA has been formed. So we have come to the conclusion that the DNA replication takes place. This is the DNA replication, and recoiling, we have recoiled it. We have recoiled it. Now it is over. RNA primer has been thrown out. Recoiling is over. Now he will take the charge of this. So in office, what do we do? Some tenderer comes and they does the work, and after completion of the 5 years, so nucleus will sign. If there is error or this and that, they will sign, yes, work is over, satisfactory. The work is satisfactory. They say that it was in the form of helix, you make the helix. So this helical form is restored and it goes away. So DNA replication is done. So this is done in million and million of cells. So we are learning it, and this is done in this way. It is not that much difficult. I think it has given you a little bit knowledge about the DNA replication. Whatever I have said, you just take a note of it and rearrange it as per your convenience, so that in examination hall you may divert from this time, you make a simple note, say about for 30 minutes or 20 minutes notes, and then you write it. Thank you very much.